Hello everybody, my name is Navonita Rai Chattopadhyay.
I am working as an assistant professor in the Department of Biotechnology, Haldia Institute of Technology. Today I will be discussing a few basic things about biology which should be known to every person. Basically, I will be discussing what is a gene, what is DNA or RNA, what is the relationship of a gene with chromosomes, where chromosomes are found inside our body, and so on. All multicellular eukaryotic organisms like human beings are made up of millions and millions of cells. Each and every cell has a nucleus which contains chromosomes. Now, chromo means color and soma means body. That's why the names are called chromosomes, as they can be seen as stained bodies under microscope. Now, chromosomes contain the genes. Usually, we know that genes are responsible for all the characters which can be seen inside us, and chromosomes carry the genes. Actually, chromosomes are nothing but a long molecule of DNA double helix which is packed with the help of various types of proteins. So, if you can remove all the proteins from one chromosome, you can get one long DNA double helix from one chromosome. Say for example, in human beings, there are 46 chromosomes in each cell. Therefore, if you can remove all the proteins from all the chromosomes, you will get 46 DNA molecules from each human cell. I will discuss how these DNA molecules are packed to form chromosomes. But before going to the details of that part, I will discuss how DNA and RNA molecules are made. The full form of DNA is deoxyribonucleic acid and the full form of RNA is ribonucleic acid. These two molecules are needed for carrying the characters from one generation to the next generation and for expressing all the characters which can be seen in us. Now, DNA and RNA, these molecules are nothing but polymers of nucleotides. Now, what are nucleotides? Nucleotides are nucleosides plus a phosphate group, PO4 group. So, what is a nucleoside? A nucleoside is a ribose sugar and a nitrogenous base attached with this sugar. So, a nitrogenous base and a sugar, these two makes the nucleoside, and when a phosphate group is attached to it, then it will be called as a nucleotide. Now, DNA and RNA molecules are long polymers of these nucleotides. Ribose is a 5-carbon sugar. Each of those carbons are numbered from 1, 2, 3, 4 and 5, and each of those carbons valencies are completed with the help of one hydrogen and one OH radical. In RNA, the ribose sugar is used, but in DNA, deoxyribose sugar is used. The second carbon of deoxyribose sugar lacks one oxygen from that OH group. Therefore, the second carbon in deoxyribose sugar contains two hydrogens to complete its valencies. So, in DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. Now, the phosphate group. Phosphate group PO4 3- that is attached to each sugar moiety. The fifth carbon accepts the phosphate for making nucleotide from nucleoside. Nucleotides contain 3 phosphate groups attached one after another. During the making of the polynucleotide chain, that is the nucleic acids, DNA or RNA long chains, 2 phosphate groups are removed. Only one phosphate group remains as the integral part of each nucleotide. Now, the nitrogenous bases. As I always tell that each and every biological term has a meaning. Nitrogenous bases are molecules which contain nitrogens in its integral ring structure. And by nature, they are basic. They are not acidic molecules. In DNA and RNA, 2 broad categories of nitrogenous bases are used, purines and pyrimidines. In case of purines, adenine and guanine, these two are used for making both DNA and RNA. In pyrimidines, 3 nitrogenous bases are there, thymine, uracil and cytosine. Now, cytosine is present in both DNA and RNA, but uracil is present in RNA, and instead of uracil in DNA, you can find thymine. During making of the polynucleotide chain like DNA or RNA, as a rule, the first carbon of the ribose sugar will be joined with the nitrogenous base, either adenine or guanine or thymine or uracil or cytosine. The fifth carbon contains the phosphate group as free nucleotides, and during the making of polynucleotide chain, the third carbon will be joined to the phosphate group of the fifth carbon of the next coming nucleotide. So the phosphodiester bond is formed in the third carbon of the ribose sugar. Here in this image, you can see that two hydrogen bonds can be formed between adenine and thymine, and three hydrogen bonds can be formed between guanine and cytosine. Therefore, as a rule, when the DNA double helix is made, adenine always pairs with thymine and guanine always pairs with cytosine. Where the DNA double helical structure is shown, you can see that if you can determine the sequence of adenine, guanine, thymine, cytosine, etc. in one strand, you can easily determine the sequence of bases in the other strand. And sugar is common to all the nucleotides. Phosphate is common to all the nucleotides. Therefore, if you know the sequence of those nitrogenous bases in one strand, you can easily determine the sequence of nitrogenous bases in the other strand. Now again, in this image, in the left hand side panel, you can see that a single strand is shown, and in that image, red circles are made in one oxygen atom for each phosphate. During the process of making of DNA and RNA, phosphodiester bonds are formed between the fifth carbon and the third carbon of two nucleotides. One oxygen remains free in that type. In normal physiological condition, the OH group has a tendency to release the H plus from it. So the OH group of the phosphate actually remains as O minus in such cases. As we know, if a molecule has a tendency to release H plus ions from it, it should be called as an acid. That's why the name is ribonucleic acid or deoxyribonucleic acid. Here in this image, I have just shown the simpler diagrams of DNA and RNA and the models which can be found for single-stranded RNA molecules and DNA double helical molecules. Now I will discuss the polarity of DNA or RNA. Previously, as I was discussing about the formation of the polynucleotide chain, I have told that the third carbon of the first nucleotide will form the phosphodiester bond with the fifth carbon of the next coming nucleotide, and likewise the chain will be made. So at the beginning of the nucleotide chain, you can find one phosphate group attached with the free 5' carbon, and at the end of the nucleotide chain you can find one free OH group, and that makes the polarity from 5' to 3'. Whenever a DNA double helix is formed, it should be thermodynamically very stable. The first rule for this stability is adenine will pair up with thymine and guanine will pair up with cytosine. The second rule is the polarity of these two strands of DNA must be opposite to each other. As you can see in the image, they are shown with arrows. By convention, whenever you are drawing two DNA strands, the upper strands will start from 5' polarity and it will end up with 3' polarity. But if you think about the interior of a cell, there is no upside or downside. You cannot say that a molecule is present as upside or it is present in downside, or a strand is present upside or downside likewise. So if you want to draw the opposite thing, that is from 3' to 5' and the next strand is from 5' to 3', you can draw that. But by convention, if you are not mentioning the 5', 3' numbers, the upper strand represents the 5' to 3' polarity and the lower strand represents the 3' to 5' polarity. As most of the RNAs are single-stranded, they are always 5' to 3'. And now I will discuss how DNA is packed in chromosomes. Average diameter of a DNA double helix is about 2 nanometer. Now this double helix is packed by the help of histone proteins. There are 4 types of histone proteins which are used for packing the DNA, and these 4 histones are H2A, that is histone 2A, H2B, that is histone 2B, H3 and H4. 2 molecules of each of these histones are used to make a core which is called a histone core. Now DNA is wrapped around that core. Near about 146 to 147 base pairs of DNA is wrapped around twice around that histone core, and this total thing is called as a nucleosome. First, the 2 nanometer DNA is wrapped around histones, and the diameter of that string will become about 11 nanometer. And this structure is also called as a beads on a string structure, because you can see this structure in transmission electron microscopy as just like beads on a string. In the next round of packing, this beads on a string structure is packed just like a spring, and this is called a solenoid structure, and the diameter, average diameter of such type of structure is 30 nanometer. This 30 nanometer string is normally seen as the interface chromatin, and we call these things as chromatin during our experiments. Now, by the help of some non-histone proteins, this 30 nanometer string is packed in some loops, and the average diameter of that structure is 300 nanometer. Then again, this 300 nanometer structure is packed as a condensed loop, and the average diameter will be 700 nanometer. Finally, these super condensed 700 nanometer structures will make the 1400 nanometer structure of chromosomes, which can be seen as metaphase chromosomes under microscope. This chromosome with 1400 nanometer diameter contains 2 sister chromatids. So 2 DNA molecules, replicated DNA molecules attached with each other and condensed, that structure is seen as 1400 nanometer structure in metaphase stage of cell division. Now let me tell you a few things about those histones. As I was telling, 4 histones and 2 molecules of each of those 4 histones are needed to make the histone core, and that is also called as a histone octamer. 146 to 147 of base pairs of DNA is wrapped around twice around this histone core, and this total structure is called a nucleosome core. Now, from one nucleosome to another nucleosome, there is a string of DNA, and that is called the linker DNA. First of all, depending upon the species, the length of the linker DNA may vary. But the length of the DNA which is wrapped around the histone core, that remains almost constant for all the species. So the length of linker DNA is very important for determining the species. Another histone, histone 1, that is also needed for stabilizing the nucleosomes. One molecule of histone 1 is needed for stabilizing that DNA double helix which is wrapped around the nucleosome core. Now, what is a gene? Gene is nothing but a part of DNA. That particular part can make some protein. That particular part can make some functional RNA. As I have told previously, that if you can remove all the proteins from one chromosome, then you will get one long strand of DNA double helix. So parts of that DNA double helix are called as separate genes. So in one chromosome, there will be a specific position or locus for a particular gene. Usually in eukaryotes, the chromosomes remain as pairs, and each member of that pair of chromosomes contains same genes at the same locus. But the alleles of the varieties may be different. Say for example, in human chromosome, suppose one chromosome pair contains the gene of height. So the name is gene of height, but one member of that chromosome pair may contain the tall variety of gene of height, and the other member of that pair may contain the short variety of the gene of height. So that's all for today. If you have any question, you can ask me in the comments. If you like my video, just press the like button and subscribe the channel. Stay safe, stay healthy. See you soon. Bye.